many pathogens are able to get past our external barriers and enter our body. The microorganisms first encounter nonspecific defenses, including phagocytes like macrophages. Then the specific immune response of T cells and B cells begins. T cells and B cells recognize an antigen through antigen receptors bound to their plasma membranes. Each T or B cell has about 100,000 antigen receptors. All the antigen receptors on the same cell recognize the same antigen. Only lymphocytes that have the antigen receptor for a particular antigen are activated by the invader. This lets the immune system respond to each specific pathogen independently. But how can we defend against different pathogens if each lymphocyte responds only to a particular antigen? The answer lies in diversity. During embryonic development, immature T and B cells undergo a type of genetic recombination that produces a single functional gene for each polypeptide of the antigen receptor. The recombination is unique for each cell. The huge pool of T and B cells, each with a unique antigen receptor, can therefore respond to millions of different antigens. Before T and B cells can be activated by an antigen through their antigen receptors, the antigen must be presented in a way that the cells can recognize it. This is the job of the molecules of the major histocompatibility complex, or MHC. The MHC is a set of cell surface molecules encoded by a group of related genes called a gene family. There are two classes of MHC molecules. MHC1 molecules are found on the surface of all cells. MHC2 molecules are found only on the surface of specialized antigen-presenting cells, including macrophages and B cells. The specific immune response begins when a lymphocyte recognizes that the MHC molecule or the antigen it is presenting does not belong to the body. Once a particular T or B cell is activated by an antigen, it begins a series of cell divisions called clonal selection. Clonal selection produces two groups, or clones, of cells. One clonal set of cells consists of short-lived effector cells, which work to destroy the specific antigen. The other clonal set of cells consists of memory cells, long-lived cells that have receptors for the specific antigen. The clonal selection of specific lymphocytes that occurs when we are first exposed to an antigen is the primary immune response. It takes about 2 weeks to generate the maximum effector cell response. During this time, symptoms of the illness may appear, like the spots from chickenpox. As the primary immune response proceeds, the pathogen is destroyed and the symptoms eventually disappear. If we are exposed to the same pathogen again, the secondary immune response is much faster, taking only 2 to 7 days thanks to the memory cells. The memory cells divide rapidly and produce more effector cells when they encounter the pathogen again. Since the secondary response is so fast, we generally don't have time to develop the symptoms of the illness before the pathogen is destroyed. Because of memory cells, we rarely get chickenpox or many other illnesses twice. Now that you're familiar with the components of the specific immune response, let's look at the cellular response of T cells and the humoral response of B cells and antibodies.